growing all over India. The leaves and bark of this plant is used Moravia diffusa. That is also one of the important medicinal plant which is growing all over the world, and the entire plant it is used as a medicine. Then shatavari, that is asparagus racemosus, that is growing through the tubers. If you grow once, then it is spreading all over the area center from where it is growing. So it is growing for years together. Therefore, it is described perennial. It grows on the another plants like a climber, grows up to 1 to 2 meters, and found widely growing throughout the India, and for the purpose of medicine. This plant is also cultivated commercially. Then shigru or moringa olifera, or that is known as seoga. So this is also one of the important medicinal plant, of which the leaves, bark, fruit, flower and seed. This is used as a food, and this is one of the very important plant which is known as moringa or drumstick tree, which grows up to a height of 20 to 40 feet. And these pods of this plant that is used very commonly as a vegetable, and it is also one of the very important medicinal plant. Then shuthi, that is ginger officially, or it is known as adrak, which we use in our day-to-day life as a one of the very important component in the kitchen to prepare the different vegetables, etc. So this is a underground modification of the root in the form of ginger. So it is having high medicinal use, and it is one of the very important spice, and that is also known as folk medicine. This is growing herbaceously, and this stem, that is the underground, that will convert into a rhizome-like structure, which is one of the very important medicine. Then tulsi, Ocimum sanctum, Ocimum tenuiforum, number of these species are there. But in general, this tulsi, everybody knows it. That is also known as holy basil. It is growing all over the world. It is growing wildly without any cultivation, plantation. But for the purpose of medicine, for the purpose of ornamentation, for the purpose of beautification of the home, or in order to worship, this is one of the important plant which is daily worshipped, and this is also having a very important commercial or our day-to-day life use as a medicine. Just a decoction of this tulsi is very important to cure the cough. Next one is the Vasa or Justica adathoda, or now it is known as Adathoda vasaka, or that is known as Adulsa. So it is a hedge plant on the boundary of the home, field. Widely it is growing, and it is one of the very important syrup. In the form of syrup, it is given to the children from the fever or another number of the diseases. It is also a widely growing plant. Then last one is the Apis precotarius, or it is known as gunz, or in the betel while eating we used to eat the leaves of these plants. This entire plant is very useful. These not only it is used as a medicine, but the seeds of this fruit are very beautiful, or the strings are made, and a single seed it is also used as a wet. And these tribal people, they used to use these seeds or a string of a seed as a ornament. So these are some of the important medicinal plants. So first we try to see what is fungi. Fungi means it is a Latin word of which the meaning is mucus, or it is like mushroom. So I think you know very well the mushroom. That is the edible fungi is known as mushroom. The fungi which are eaten as food are known as mushroom. The fungi which are not eaten as food or which are poisonous are known as toadstools. So these fungi, these are macroscopic as well as microscopic. So during any visit to garden or the forest, you might have seen these fungi growing on the stem of the plant, leaf of the plant, inside the plant, outside the plant. These fungi, if you see under microscope, these are unicellular or these are filamentous, and this vegetative part of the body of the fungi is known as mycelium, and a single part of the fungi is known as hypha, and in plural that is known as hyphae. So many number of the hyphae made mycelium, or mycelium consists of many number of the hyphae. So these are unicellular, multicellular. They are completing their life cycle as a saprophyte, or the saprops means those fungi which feed on dead tissue or organic waste, and simultaneously they cause the process that is known as decomposition, where these substrate and the fungi both are get benefit from each other. They are converting this complex material into simpler and absorbable form. Those are known as decomposers. Then the next one is the symbionts, where they mutually get benefited from each other. That is, they forming a beneficial relationship with the another organism, and there is no any harm to each other. And last one is the parasite, which feed on living tissue of a host. Host is the living organism on which the fungi grows. So these are certain general characteristic fungi of these in the nature. So these are again different important characteristic features. If you just come to know what are the fungi, that is also sufficient. Then the just now I have described that a single part or segment of the fungi is known as hypha, and plural is the hyphae. Here you see the unicellular mycelium, or you can say there is no any partition wall in between the mycelium, that is known as chinocytic hyphae. First one, where you can see this mycelium, it is divided into number of the cells due to a partition wall, and that is known as the septate hyphae. First one. Second one is a cenocytic, or that is also known as a septate hyphae. And the third diagram, where you can see these the yellow color is the fungus, and this square, that is a green color, is the plant cell, where you can see this from the hyphae of the mycelium, these fungi, it is entering into the getting the ready-made food material from the plant cell, and it is growing, reproducing, and causing the plant diseases. So another example, it is not only causing the damage to the host plants, but it is also causing examples or group of the fungi which are forming this association known as mycorrhizae, that is the zygomycotina, scomycotina, basidiomycotina, etc. Then in the such a manner, same another example of forming the fungi association with the another living organism that is known as algae, or which is wildly growing on the surface of the water, inside the or at the bottom of the water, that is commonly growing and commonly known as seaweed, or in Marathi we describe it as shevala. So these fungi, they also form a symbiotic association with the algae, and that association is known as lichen. And these lichens are of three types: crustose, foliose, and fructicose. So this is also one of the very important widely growing fungi in association with algae, and this is a just like a papad, and which is made in the form of powder, and which we use as a condiment or masala in day to day our different article preparations. Then the reproduction, how it occurs? It is occurs by the structure that is known as the spores, and that is occurring asexually as well as sexually. It is occurring sexually by meiotic division and asexually by mitotic division, or directly from the part of the mycelium, that is hypha. It may be open, or it may be in a structure that is known as sporangia, or it may be just like a fruiting body. So here you can see the first pink color, that is a fruiting body, and last one here you can see the reproductive structure in an open form, that is a example of penicillium. So these are certain reproductive structures. Then in general, how the life cycle of the fungi, it is completed asexually, mainly by sexually. So this mycelium, which is undergoes the process of germination, produces the spores, and then after spore producing structures, again they undergo the germination to produce the new fungi, where only the single celled, that is haploid structures are produced. Again at the time of reproduction, fusion occurs to produce diploid, again it undergoes rest, and again from which haploid structures are produced, from which new mycelium or fungi develops. In the same way, sexual reproduction, where these main fungi, that is the undergoes the fusion, cytoplasmic fusion, that is known as plasmogamy, then nucleic division, that is known as gametogamy, and a zygote or a resting structure develops that undergoes the germination, and after the germination by meiotic division, again the diploid stage develops. So these are in short two main type of the reproduction in fungi, and there are four different classes of fungi, you can say, that is Ascomycotina or Ascomycetes, where these reproductive structures, they are sac-like, and in general the reproductive structure that is produced, it is known as ascus. It is a very common fungi, and example is the yeast, Saccharomyces cerevisiae, and this yeast, that is a very good example. It is in the preparation of the breads. These yeasts are used, and also it is also causing some of the diseases that we will see in the next part. Second one is the Basidiomycotina, or it is also known as a club fungi. So this is one of the important class of which some of the very important member of the Basidiomycotina or Basidiomycetes is the mushroom. Mushroom is the example of the class of fungi Basidiomycetes, that is eaten as a food. And in addition to this, another number of the examples are there which are causing disease, rust, smuts, etc., which are the plant parasites. Then Ditromycotina, or it is also form phylum fungi imperfecta. So these are the fungi where these are many in number, and they are causing many diseases to the foliage parts of the plants, and it is not showing the entire characteristic features of the different categories of the classification known as the imperfecta. The stages, some of the stages are absent, or the life cycle is not complete. Therefore, it is known described as form, class, form, order, form, phylum, etc. Therefore, it is described as imperfect fungi. So that is about the fungi, structure, reproduction, and what is the use of these fungi. And we have also seen some of the fungi are beneficial fungi, some are those are causing decomposition, biosynthesis, those are acting as a drugs, antibiotics, alcohol, acid. They are useful in the food processing, that is fermentation. Those are model organisms for biochemical and genetic studies. Some of the fungi are harmful. Some of the fungi are destructive in food, paper, etc. They are causing animal, human diseases. They causing allergies. They are producing the toxins. uh, uh, Good example of the toxin, it is the aflatoxin produced in the groundnut, etc. But our main concentration is on the last point, that is the plant diseases. We will see some of the important crop plants and the diseases caused by the fungi. So this is the field of the turmeric, that is curcuma longa. So it is cultivated nowadays on large scale. So this is the plant where you can see very clearly, as the overground part of the plant grows, it produces the leaves. Leaves becomes larger. It receives maximum solar light, and with the help of chloroplast and water, it synthesizes the food material, and whatever the food material that is synthesized, it is transported down a root, it is converted into the tubers, and that is the important commercial part. Here you can see one of the elaborated leaf of the turmeric plant, where you can see in a restricted part there is a killing of the host has been occurred, and it is one of the sample, but it may also occur on large scale, and near about 20 to 30% yield loss, decrease in the production of the turmeric occurs due to this fungi, and therefore it is considered for the study. So how to study this? That is this much infected part, it is placed on the petri plate, which is having the nutrient or the food material to grow the fungi, that is known as media, that is potato dextrose agar media, that is prepared with the help of autoclaving, that is containing 20 gram of the potato peels, 15 gram of the dextrose, 20 gram of the agar. It is boiled and autoclaved. This autoclaving, it is nothing but just like the pressure cooker, and after that this infected part is kept, and that is allowed to grow. After a few days you see it grows, it is transferred many times, and unless and until we will get such a plate. So this fungi which is responsible for causing leaf spot of turmeric, it is identified as Colletotrichum capsicum. Throughout the world, it is only known by a single name. So it can be identified on the basis of morphological characters, reproductive characters, or nowadays by the help of rDNA technology, we can identify these fungi from the different sources like Agarkar Research Institute, etc. So we have grown this fungi from the leaf that we have collected from the field. But whether it is correct or not, whether it is responsible for causing again leaf spot or not, that can be proved by the test known as the pathogenicity test. So such type of the fungi, in the center you can see a white disc is there, white rounded part that must take out, place in a test tube, crush it with a sterile distilled water, and apply with the help of brush to the upper part of the leaf. So the second diagram here you can see a petri plate is there, a rod is placed on that. The leaf is kept, of which the petiole is only touching to the filter paper, and sterile water is added time to time to maintain the moisture in the petri dish, so that here maximum environmental condition, natural conditions are tried to provide. To one leaf, such type of the fungal suspension is not applied. To the right side, where you can see the fungal suspension is applied, after 3 to 4 days, you will see the host killing has been started. That is means this fungus is growing on the leaf. It grows first on the leaf, after that it enters into the cell of the host. Earlier I have shown you the diagram, and that is nothing but the host and pathogen interaction starts. Host means here the turmeric leaf. Pathogen means this fungi, Polytrotricum capsicum, which is entering into the host. It is interfering with the physiology of the host. Host itself has synthesized the carbohydrates or these proteins, etc. And this fungus, it is absorbing ready-made, and with that the killing of the tissue occurs. That much tissue it is reduced from synthesis of the food material, means ultimately. So this is the same under field condition. So in the beginning I showed you, on the leaf, but if it is severe, intensity of the disease increases, then the entire plant killing may also occurs. Then in the underground part, here also you can see this first diagram where there is complete decomposition or infection of the fungus has been occurred to the rhizomes, and the color has been changed and it is empty, so not having the important contents of the turmeric. Then the fungi that is responsible for underground killing of the turmeric, that is the known as Pythium myritorium, means the first rhizome which you have seen, just a small part you plate on the petri dish in the laboratory. Such type of the growth can be seen, and it is identified. It is pathogenicity is proved, and then after it is declared, it can be declared that it is due to the rotting of the rhizome. So under microscope, if you see, that is the vegetative part of the fungi, that is known as mycelium. Then these are the reproductive structures known as sporangia. Then in the earlier also I shown that pathogenicity or a disease is caused to that particular fungi. It is proved on the leaf. It is on the rhizome. So just to take a slice on which you can grow the fungi, and at the end you can see there is complete destruction, deterioration, spoilage of the rhizomes of the turmeric due to growth of this fungi. And some of the experiments, those are carried out to control by using the plant leaf extract, that is Triodex procumbens, or that is in Marathi known as Tantani, where you can see if its concentration is increased, the fungal growth is decreased, means the disease also decreased. The another plant, Ocimum sanctum. Then the another plant, that is Adathoda zhelenica, or that is known as Adulsa. It is one of the very important cough syrup producing plant, widely growing plant. So this is the flowering peak of the plant. It is widely growing, and such infected leaves are collected. So how to study a disease severity index, or how to measure the disease? So collect 100 leaves, classify into 4 grades for the purpose of the study. So here you can see 1, 2, 3, 4, 5. bundles of the leaves. How these are made? First, you can see there is no infection. Zero grade. First one, 1 to 25. Second, 26 to 50. Third, 51 to 75. And last one is 76 to 50%. And by using the formula, that is disease severity index, the disease intensity or percentage can be determined. So the pathogenicity, again, the leaf spot diseases of the adulsa, it is studied. Here you see leaf spot, a restricted part of the leaf, it is infected by fungi. Here, unlimited or larger part of the leaf, it is infected. Here, the enlarged part of the leaf spot. So earlier also I told you, if the environmental condition and the host gives the response, then the disease increases and there is the destruction of the leaf, or that much is reduced from the process of photosynthesis. The same, that is the growing the fungi in laboratory condition on petri plates. It is known as Alternaria alternata, where the visually also you can identify the disease if it is a single spot and there are certain circles of the spots around which there are certain circles of yellow ring, then that is nothing but the reproductive structures which are produced in a chain, and it is a very common fungi on almost all the plant parts you can see it. Then another species, Coledotrichum capsicii, it is a very common fungi. Then the third species, Coledotrichum demasium. Then these are the different fungi which are isolated from the disease caused by the different fungi on the leaf that I have shown to you earlier. And the next plant is the Saped Musli, that is a Chlorophytum borealum. So earlier, many of the plants, those are wild, but presently, for the purpose of the cultivation, these plants are used, and it is also one of the very important tonic cultivation. Here you can see. So in the pot also commercially nowadays to show to the students, etc., also cultivated. Then here you see the infection on the leaf. So here under severe infection, here you see the infection of the Coledotrichum capsicii. So with that, that is about the different crop plants and their diseases. So I have tried to enlighten some of the very important cultivated crop plants. But in all, there are a number of the crop plants which are nowadays used for the purpose of cultivation. Thank you. Topic is over. If anybody want to any query, questions.
Hello, sir. Hello. Hello, sir.
Did you getting me?
Hello.
क्रांत जी आवाज आ रही है आपकी.
Hi, sir. What is DNA and RNA?
Ribonucleic acid and ribonucleic acid. My speech is over, sir.